go ahead and get started recording and skip down a little bit. We're going to go back to where we were, which is we were just beginning to think about meristematic tissue. So remember, we were talking about apical meristems, and the apex of something is the tip. So we're talking about the growing tip of either a root or a stem. Now keep in mind, a stem might put out branches, so you could have an apical meristem at the very tip of a branch growing out of a tree, or at the tip of a large root. Now the key to apical meristems is they contain cells that are actively dividing by mitosis. So one way to think about this is when a cell divides by mitosis in, say, the human body, normally what happens is the cell grows and gets larger. And then when the cell divides by mitosis, when cytokinesis happens, the two daughter cells are about the same size as a normal regular cell. Why are they the same size as a normal cell? Because the parent cell grew before it divided. But in the apical meristems of plants, what we see is the cells are dividing so quickly, they don't have time to grow between mitotic divisions. And since they don't have time to grow between mitotic divisions, the cells are small. Now, if we were looking at this under a microscope and we were looking at this at low power, we wouldn't be able to see the cells very clearly. But the two parts of the cell that usually absorb the stains that we put on our microscope slides, if we're looking at a plant, the two parts are the cell wall. And usually the thicker the cell wall, the more stain it will absorb. So when we see cells that look like they have a dark edge around them, those are cells that have thick cell walls. And the other part is the nucleus. So notice when we look at the apical meristem of the shoot, the dark purplish color here is almost solid up in the apical meristem. And the reason that it's almost solid is because these cells are very tiny. They're dividing, and when they divide, they make other tiny cells. But notice as you get further down from the shoot, these are older cells that this apical meristem made. The apical meristem is constantly moving upward in the shoot tip. If we look at these cells, the little purple dots are spread out. They don't form a solid block, because one of the things that happens is after the cells are produced, they get nutrients from the rest of the plant, the parts that are carrying out photosynthesis. They grow and they get larger. And as they get larger, now the nucleus in each cell is a little bit farther apart from the nucleus in a neighboring cell than it was when it started out. Now, we can't see the same thing as clearly when we look at the apical meristem in a root, just because it turns out that in a root, cells keep dividing for a couple of cell layers back. You might remember in Biology 105, one of the things we looked at was some slides of mitosis. And the slides that we looked at of mitosis were actually slides just above the apical meristem in an onion root. And the reason we looked at those was because those cells are dividing very quickly by mitosis. So you can see all the different stages. You can see prophase and metaphase and anaphase and telophase, and you can see some cells that are between divisions that are in interphase. So let's look at these meristems in a little more detail. And notice these are apical meristems. Remember, we actually have three types. We also have lateral meristems, and we have intercalary meristems. So we'll look at those as well. So let's look first at what we see in a root. Oh, so here's a root with a diagram so we can identify some of the parts. Notice that our real root doesn't go up nearly as far as the diagram does. So they've drawn a little circle around the area that represents our apical meristem. And this is basically an oval region. Now, in this area, we have cells dividing by mitosis. And when the cells in this meristem region divide by mitosis, one cell, one of the daughter cells that's made in mitosis, stays in the meristem. So that means it can divide again later. The other daughter cell, because remember, mitosis makes two daughter cells, they're genetically identical. The other daughter cell either gets pushed downward into this thing called the root cap, or it gets pushed upwards into this thing called the zone of cell division. So sometimes when these cells in the apical meristem divide, they make another cell in the zone of cell division. Sometimes they make another cell in the root cap. Now, if we look at our real root, the real root cap is over here. And especially if you look down at the bottom, notice that there are gaps between some of the cells. We don't think of plants as actively moving through their environment, but keep in mind the root actually pushes through the soil. And as the root pushes through the soil, the root tip is on the end, there's friction. And because of this friction, the cells in the root cap get torn apart. And so the cells in the apical meristem constantly have to be pushing some new cells into the root cap, because as the root grows through the soil, this root cap protects all the cells behind it. Now, this also pushes some cells back into this zone of cell division. And the name gives you a pretty good idea of what's happening here. In the zone of cell division, cells are dividing by mitosis rapidly. So that's why when we look here right above the apical meristem, we don't see those purple dots that represent the nucleus. We don't see those spread out a lot further because we're still in that zone of cell division. Now, as we go up a little bit further, we get to the zone of elongation. It's often called the zone of cell elongation. And in the zone of cell elongation, notice that, let's magnify our view a little bit more here. If we look at the zone of cell elongation, notice that these yellow cells and these purple cells, we can probably see it most clearly in them, but also a little bit in the blue cells on the side. These cells are getting longer from top to bottom compared to the cells in the same areas that are right above the zone of cell division. So this elongation, this is really what pushes the root through the soil, because as these cells get longer, they push all the rest of the root below that point further. So that's what creates that friction that kind of tears that root cap apart eventually. Now, notice that in the zone of cell elongation, the cells are still kind of generic cells in some way. They don't start to become fully differentiated until they get to the zone of cell maturation. And in the zone of cell maturation, then they become specialized. Now, notice in a root like this area in the yellow represents the cortex. And the cortex in plants is always going to be mostly parenchyma cells. So we'll look at those in a little bit. The blue cells along the sides, some of them stick out and form root hairs. These are part of the dermal tissue. These are mostly epidermis cells or epidermal cells that are being shown. And then the purple in the middle, this is called the vascular cylinder or the vascular stele. This is where the xylem and phloem are, the vascular tissue. So when we look at our root, we can see that we have to have cell division for the root to lengthen. And some of that cell division is going to make the root longer. Some of it's going to form the root cap, and the root cap is always about the same size because it's constantly getting torn up. So it doesn't really enlarge throughout the plant's lifetime. whereas the root gets longer and longer. Now you might ask, why does the root have to grow through the soil? Why can't it just stay where it is? Well, really two reasons. First of all, as the plant gets larger, it needs more surface area in its roots. Remember when we talked about surface area a few weeks ago, the more surface area you have, the better you can either absorb or eliminate materials. In the case of the roots, they're absorbing materials. They're absorbing water, any minerals that are found in that water from the soil. And these are going to be the minerals, the nitrogen that's absorbed. This is going to be what's used to build things like proteins that contain nitrogen. Magnesium ions can be absorbed. Magnesium is part of chlorophyll. So those ions can be used to build chlorophyll. There's another reason why the plant roots have to grow. Think about if you lived in one place and you didn't have a grocery store and you could only get your food by what we call hunting and gathering when we study human societies. In hunting and gathering, you go out and you kill animals, you might trap them, you might shoot them. And in gathering, you go out and you collect fruit and stalks and berries and whatever is edible in the area. The problem with that type of lifestyle is eventually you've collected most of the edible plants in an area and you've captured most of the animals you can capture. So you're going to start running out of food. So when a root is absorbing minerals from some area, after a certain point, it's absorbed most of the minerals that are there. So it doesn't necessarily benefit the plant to continue absorbing from that same region. So what's one solution? Have the root grow. And when it grows, the root enters new regions where it hasn't tapped into the minerals yet. Water is usually replenished on a pretty regular basis, so the roots don't have to grow too much to give the plant access to more water. So the apical meristem on the root is crucial. We can also look at the stem tip in a plant like Coleus, and I forgot to italicize the genus name. And this also produces an increase in the length of the plant. So here's an enlarged view of that Coleus that we saw earlier. And as these apical meristem cells divide, and again, notice you can see that the purple dots are closer together in the meristem than they are down here or over here. As these cells in the apical meristem divide, they produce cells in this region. And these cells, notice as we go towards the bottom, gradually get longer. So we don't really call this the cell of elongation in the zone of elongation in a shoot, but it is very similar. Now, right behind the meristem where it's growing, every once in a while, we see new leaves start to form. And these new leaves are called a leaf primordium. Primordium means beginning. So notice in the leaf primordium, again, the cell nuclei are very close together. Down here is a leaf that was a primordium when the apical meristem was down at the bottom of this area. And now this leaf is older. And notice that when you look at purple dots, they're much further apart because this leaf has grown, the cells have elongated. Now, right above these older leaves, you can see another cluster of very close cell nuclei. And this is a lateral branch bud. So this is where a little branch is going to come off. Don't just think of a branch like you might see on a tree. That would be one possibility, but Coleus isn't a tree and it forms branches. A branch could be something like a side branch that you have on a tomato plant. So this is going to be the beginning of that side branch. When we look at buds on trees in the spring, there are a couple of types of buds. Some of them are lateral branch buds, but some of them are actually leaf buds or flower buds. Just depends on the case. And that's going to be the first growth for that plant when spring comes along and the growing season begins again. So apical meristems, if you would pull a plant up and you would measure it from the tip of the shoot to the very tip of the root, apical meristems make the plant longer from one tip to the other. And the first xylem and phloem and every other type of tissue that a plant makes comes from these apical meristems. You can see these apical meristems in a seed. If you have peanuts at home, take a peanut and open up the shell, break a peanut in half very carefully. And if you look on one half of the peanut, you can see a little structure that looks kind of like a carrot. The part that looks more like the carrot is the beginning of the root for that baby plant. The part that looks kind of like a couple of little leaves is the beginning of the shoot for that plant. So all the parts of the plant that are produced from the two apical meristems and from the original embryo, this is called primary growth. And we know, of course, that primary means first. But when we look at lateral meristems, lateral means side. Lateral meristems are towards the side of a plant, and lateral meristems make the plant wider. Or if you're thinking in mathematical terms, they increase the diameter of the plant, or the circumference, or the radius. You could use any of those terms. Lateral meristems produce something called secondary growth. And secondary growth doesn't make the plant longer from root tip to shoot tip. Secondary growth makes the plant thicker. So let's look at a diagram of a tree, and let's peel out the different layers. So let's start first with this kind of bluish layer, which notice they peeled off, but really goes all the way around the plant. This is the vascular cambium. This is one of two types of lateral meristems. And cells in the vascular cambium divide by mitosis. And again, when cells divide by mitosis, there are always two daughter cells. So one cell stays in the vascular cambium and it can divide again. The other cell either gets pushed towards the middle and forms this light purple area. And this light purple area is called secondary xylem. Remember, we just said that lateral meristems produce secondary growth. Or when a cell in this vascular cambium divides by mitosis, it can push one cell towards the outside where it forms this light green, and this light green is the secondary phloem. Now notice this vascular cambium, when you look at an actual tree trunk, can be very difficult to see because it's usually one or two cells thick. So it's not very thick at all. So let's think about our tree. Now, when the tree first starts growing, the beginning tree doesn't really have any wood yet. So this vascular cambium hasn't started making secondary xylem or secondary phloem. So let's think about just starting with this orange. When the tree seed first germinates, we have this area in the middle, which is the pith. And then we have a band of dark purple around this pith. And this dark purple is the primary xylem. So this is the first xylem that was made. And then if we don't have any secondary growth yet, if the vascular cambium hasn't started doing anything. We don't have any of this light purple. We've got a thin layer of blue. We don't have this light green. So right after the blue, we've got this dark green. We've got the primary xylem. And then on the very outside, we have a layer of cells called the epidermis. So before we get wood, we have the pith, we have the primary xylem, we have the vascular cambium, we have the primary phloem. We do have some area out here after the primary phloem called the cortex. The cortex is gone in a tree, or it's been replaced, I should say. So once the vascular cambium starts dividing by mitosis, now we get these purple areas. Now, xylem has one main function in plants. It carries water and all the minerals that are dissolved in the water up. It carries them up from the roots to leaves, to fruits, to parts of the stem, whatever needs them. So in parts of the world where the growing season isn't equal throughout the year, which is most parts of the world, what happens is at the beginning of the growing season, the xylem cells that are made are very large. Why are they large? Because at the beginning of the growing season, think about the spring here in Illinois, a day like today, there's more there's more water in the spring, more rainfall in the spring than there is in the summer. Summers tend to be dry. So since there's more water, xylem cells are large. But as the growing season progresses, as we go through the summer and the weather becomes drier, there's not as much water to carry upwards. So the xylem cells that are made later in the year are smaller. And when the xylem cells are smaller, they look darker. Now we have another type of lateral meristem. We have a layer that's right on the outside of this dark green. The dark green is the primary phloem. And right on the outside of this is a layer called the cork cambium. And the cork cambium divides by mitosis. And again, one cell stays in the cambium, one daughter cell, and one daughter cell is pushed outward to form bark. And, you know, if you've ever played with the bark on a tree, bark floats on water, at least for a while till it gets soggy. Why does it float? Because it's cork. And cork cells are, like some other cells we'll talk about in a plant, cork cells are dead, and they sometimes have hollow areas in them. So these hollow areas are filled with air, so cork tends to float. So let's look at these in a little more detail on a real plant. So here's an actual tree. This looks like it's from a tree called Tilia, which is the common name is something called basswood. It's a very common tree in forests in this part of the country. If we look at this tree, here is the pith in the middle. The pith is parenchyma cells. We'll talk more about parenchyma cells in a moment, but parenchyma cells tend to be large. They have very thin cell walls. Notice this big globs of purple that you can see in here. This isn't really the cells. This is the dye we put on here to make things show up better. And there are just a couple of clumps of dye in the pith. Now, around the pith, we have the various layers of xylem. Now, one layer of xylem is separated from the next by a ring. So there's a little kind of a reddish, irregular shaped structure here. What's inside this is the first xylem that was made, the primary xylem. Outside this is the secondary xylem till we get out to here. So this is all secondary xylem. Now, if we look at this secondary xylem, how many distinct bands can you see in this secondary xylem? As you go from right here, from this kind of reddish area, all the way out to here, how many bands do you see? Should be able to see 3 distinct bands. Here's one in here, here's another in here, here's another in here. So if this tree, basswood is an example, but if this tree is found in an area where there are cold winters, during cold winters, most of the water is frozen. It's either in the form of ice or snow, so the water can't be absorbed by plant roots. In plants that live in environments like we have here in Chicago, what's called a temperate environment, where we have moist, warm summers and we have cold, dry winters. These rings show us how old the tree is. Now, if we look at these rings carefully, notice that, let's focus over here on this outer ring. So here we can see the 3 rings. Notice if we look at this outer ring, the cells towards this inner part, almost right where it says xylem ray, these cells are very big. Now, xylem cells are actually dead. The only thing that's left is the cell wall. So the cytoplasm deteriorates, all the organelles that are in the cytoplasm deteriorates, the nucleus deteriorates. The xylem cells are basically tubes, are pipes that carry the water along with dissolved minerals from the roots upward. Now, notice towards the back, the xylem cells are a little bit smaller. Since the xylem cell is dead, since everything inside the cell walls is gone, when the cells get smaller, they look darker because the only thing we can see is the cell wall. So if we have a big opening inside the cell wall, the cells look kind of light. If it's a small opening, the cells look kind of dark. So if we look at the cells that are produced early in the year, what's sometimes called spring wood, or sometimes it's just called early wood, if we look at those xylem cells, they're bigger. At the very end of the year, this little dark band that you see, this is actually very, very tiny xylem cells. So the ring that we see is when we have very, very small xylem cells right next to very, very big xylem cells. Now notice these xylem cells here that form this ring, this is actually the ring from the end of the 2nd year. Notice the vascular cambium is right in here. So this means when it makes phloem, it's making phloem and it's pushing it into the middle this way. So the primary phloem is all the way in the middle, this first band that we see here. This is, I'm sorry, I said primary phloem, primary xylem. This first band of secondary xylem here, this is the xylem from the 1st year of growth. This middle band here, this is the xylem from the 2nd year of growth. This 3rd band here, this is the xylem from the 3rd year. So 3rd year, 2nd year in the middle, 1st year out here. So notice the oldest secondary xylem is always on the outside, closest to the vascular cambium. So this tree is 3 years old. Now keep in mind, I think I mentioned before, some trees in the tropics live in areas where there are 2 rainy seasons per year, and they actually make 2 rings per year. In this part of the country, since they only make 1, these rings are often called annual growth rings, but they aren't annual in all plants. We want to keep that in mind. Now, when we look at our vascular cambium, it's a very narrow layer because this vascular cambium, remember what I mentioned, is 1 at the most 2 layers of cells. Whereas, you know, if we look at the xylem, if we would count, it looks like there's maybe about 10 xylem cells in each growth ring. Notice you can look at the growth rings, and a growth ring that's thicker indicates that that was a wetter year. So people who study ancient cultures, since many ancient cultures built, you know, some of their dwellings from wood, we can look at the wood that's in those structures, and we can get a feel for what the climate was like. We can tell if there were drought years and there were rainy years. Now, outside the vascular cambium, we see these kind of triangles. This is the secondary phloem. Now, notice the cork cambium is kind of the inner dark layer here. It's hard to make out very clearly. And then if we look at a tree, when the cork cambium starts to divide, it makes something called the periderm. Peri means around, remember perimeter from math. And derm is skin, like a dermatologist. And the periderm gradually tears apart the epidermis. And if the plant lives long enough, the periderm replaces the epidermis in a plant that has secondary growth. Now, keep in mind, if we look at the phloem and we look at this little greenish band out here, this is part of the cortex. And if we look at the periderm and what's left of the epidermis, this is the area of a woody plant that we call the bark. The xylem is the area that we call the wood. So think about a tree. If you have never seen a tree that's, you know, been cut down, if you've never looked at the stump, in a large tree that's been cut down, this pith that's in the middle might be as small as a dime. It might be as big as a silver dollar, but it's really not very big. If we look at the bark on the outside of a tree, this is maybe a ½ inch thick. So most of the thickness of the tree is xylem, secondary xylem in particular. So that's what we see in a woody plant. So here's some examples of some more tilia stem cross sections. So here's a stem that's only 1 year old. Notice that you don't see any divisions in the secondary xylem. Here's a stem that's 2 years old. You can see the division right here. So the first year secondary xylem is on this side of this line. The second year secondary xylem is on this side. Here we can see a 3-year stem. So the first year secondary xylem is in here. The second year secondary xylem is in here. And the third year secondary xylem is in here. Notice you can tell by looking at this that the second year was a wetter year than the third year, unless they cut this down in the third year and, you know, it didn't have a chance to make all the xylem it was going to. So we can still see the same basic structures, but remember what we said, secondary growth increases the diameter of a plant. So notice that the circle is gradually getting larger. We're increasing the diameter. Okay, let's see. So let's look at some of the cells we've been talking about, like parenchyma. And parenchyma tend to be big cells. They tend to have thin cell walls. And you might remember from 105, plant cells make a primary cell wall first. And the primary cell wall is relatively thin and flexible. And then some plant cells make a secondary cell wall that's much thicker and stiffer, it's more rigid. If we look at parenchyma cells, they have a relatively thin cell wall, and it's only a primary cell wall. Parenchyma cells don't have a secondary cell wall. Now, parenchyma cells do almost everything in a plant. Here we can see some parenchyma cells with some little pinkish purple dots inside them. These little dots are starch. So one of the things that parenchyma cells do is they store starch. Notice that if you would look at something like the root of a carrot, you would see something similar to this. Carrots store a lot of starch in the parenchyma cells of their roots. Now, besides being kind of large cells, having thin cell walls, the other thing we can see when we look at parenchyma cells is they're not packed together real tight. They're kind of loosely packed. So here's a parenchyma cell, here's another one, here's one. Notice this little white kind of rectangle between them. This is a space where the cells don't quite meet. You know, think of it like packing a bunch of tomatoes. Since tomatoes are round, you have some air spaces between the tomatoes. So the parenchyma cells are more oval, but we have some little spaces between them. Now, if you would look at a plant and you could just kind of reach into a grab bag of all those cells that are in that plant, and you could pull out one cell, it would probably be a parenchyma cell. In most plants, most of the cells are parenchyma cells. Here, the parenchyma cells make up a part of the root called the cortex. So we'll come back to the cortex later, but in fact, right here. So here's kind of a diagram of that same type of root. So in the very center of the plant, we have the xylem. Just outside the xylem, we have the phloem. And notice that this is a pattern that we're going to see consistently. The xylem is always inside the phloem. If we start from the outside of the plant and we were, doing a dissection or a surgery, we'd cut through the phloem before we'd get to the xylem. Now, around the xylem and phloem, there's a layer in here that they show in kind of a light gray, and this is called the pericycle. And then outside that, there's a layer that they show in a much darker gray, almost a black. This is the endodermis. On the outside of the plant, there's the epidermis. Sometimes, not in all plants, but sometimes around the vascular tissue, we have something called the endodermis. So if we have cells outside the xylem and phloem, like all these little cells out here, this whole region is called the cortex. Now, it just so happens that in a dicot root, the cortex, excuse me, is made of parenchyma cells. And then notice the very outer layer of cells, of course, is our epidermis. So sometimes from the endodermis on in, all the tissues that are associated with the xylem and phloem are called the stele or the vascular stele sometimes. So let's look. This is a root like we would see from a dicot if we focused on some of the parenchyma cells. So let's look at some other types of cross sections that we could see. And remember what we said the other day, if we're saying that something is a cross section, think of taking a structure like your arm and cutting perpendicular to the long axis. So here we're taking the long direction of a root or a stem in just a moment, and we're cutting perpendicular to that. So this is a monocot root from Zea mays. And Zea mays is the genus and species of a plant that we commonly call corn. You might know that another common name for corn is maize. So this is spelled differently. The common name for corn, maize, is M-A-I-Z-E. The species name is M-A-Y-S for maize. like Willie Mays. So around the very outside, these dark circles are our epidermis circles. All of this material in here, all of this light green is the cortex. And in a root, this cortex is almost completely parenchyma cells. Now this dark ring towards the center, this is the endodermis. And then we have these ring of cells inside the endodermis. This is the pericycle. Now let's start with these kind of large cells that look like they have 3 or 4 parts to them. The largest cells are always xylem cells. So this is xylem right in here. These more circular, lighter cells, this is the phloem. And remember something we just said, the phloem is always towards the outside of the xylem. So notice the xylem extends further in than the phloem does. Now in the middle of all the xylem and phloem here, in the center, there's an area of cells again. If we have an area outside the xylem and phloem, this is called the cortex. When we have an area inside the xylem and phloem, this is called the pith, P-I-T-H. And the pith is also parenchyma cells. So it's cells that are similar to the cells out here in the cortex. Now, if we go back to our dicot root, notice the xylem forms a kind of a cross in the middle, and then the phloem is in between the arms of this X. Notice the xylem goes all the way to the middle of the cell. So if you think about what we just said, the pith is the area in the middle of the xylem and phloem. Well, in a dicot root, there is no pith in the middle, because when we look at our dicot xylem, it goes all the way into the center of the root. So one of the things we can do, and this is kind of our starting point, if we look at the pattern of xylem and phloem in a cross section. So in a dicot root, we see this kind of X-shaped with the phloem in between the branches of the xylem. In a monocot, we see this ring of xylem and phloem. If we look at the pattern of the xylem and the phloem, how they're arranged, this is one of the ways that we can tell a monocot stem apart from a monocot root, or a dicot stem apart from a dicot root, or in this case, a monocot root doesn't look the same as a monocot or a dicot root. So if we look at the arrangement of monocots and dicots, at the arrangement of xylem and phloem, this can help us tell whether we're looking at a monocot or dicot. So we'll talk more about this towards the end, but remember the examples we gave the other day. When you think of a monocot, think of a blade of grass. When you think of a dicot, think of a leaf that comes from a tree or a bush. So let's look at the roots. The stems. Now we talked about 2 types of roots, a monocot root and a dicot root. When we talk about stems, we're going to think about 3 types of stems, because there's monocots and dicots, that's true, but there are 2 different types of dicots. Some dicots are trees, and they have that secondary growth that we talked about. They have wood. Some dicots don't have secondary growth. They're what are called herbaceous dicots, or they're sometimes called non-woody dicots. And one example of this is a plant called Helianthus. Helianthus is the scientific genus for sunflowers, and the name comes from Helios. Helios is the Greek god of the sun. So a lot of times names come from something that is vaguely familiar in terms of how they work. Okay, so if we look at this, in the middle, we have this big area inside the xylem and phloem. And so this is the pith, and this pith is going to be mostly parenchyma cells. Now, our vascular bundles, our xylem and phloem, are these clumps in here. They kind of remind me of like candy corn, like you see in Halloween, because there's a couple of different bands with different colors. If we look to the outside of these bundles, this area out here, it's much thinner than the pith. This area is the cortex. And again, the cortex is mostly parenchyma cells, but in dicots that don't make wood, there's usually another type of cell in here called collenchyma. And then our very outer layer of cells, like always, unless we have wood, is the epidermis. So our vascular bundles are arranged in a circle here. But notice this circle of vascular tissue is very different than this circle that we just saw in the monocot root. So you wouldn't look at these and get them confused. So if we look at these, the kind of purplish cells towards the middle of each clump, this is the xylem cells. The kind of greenish band in here, this is the phloem cells. And then towards the outside, this big purple clump, this is some cells called sclerenchyma. And sclerenchyma cells are mostly used to support plants, to hold them up into the light. So these plants don't have wood. Wood can help hold a plant up. They don't have a lot of xylem. Xylem can help hold a plant up. So this sclerenchyma cap helps hold the plant up. So notice we have dermal tissue around the outside, the epidermis. We have ground tissue, that's the cortex and the pith. And then we have vascular tissue, those vascular bundles made of xylem and phloem. And then there's some sclerenchyma cells associated with them. Now let's think about a monocot stem. So here's a monocot stem. Our vascular bundles are scattered throughout this. If we look carefully, we can sometimes see they're more or less in concentric circles. This is again from Zea mays, a corn plant. And as the stalk gets bigger, it might start out with one circle of vascular bundles, and then it might make another circle and another circle. So the vascular bundles that are most recent are on the outside here. Well, notice it's kind of hard to tell what's inside the vascular tissue to the inner part of the tree and what's outside the vascular tissue. So when we look at a monocot stem, we don't usually say that there's a pith or a cortex. We just call all these cells in the middle between the vascular bundles, we call all of them ground tissue. And notice that they're pretty much all parenchyma cells. There's sometimes some collenchyma towards the very outside, so right inside the epidermis. The vascular bundles are the bundles that we looked at before that were sometimes called monkey faces. So let's look at one of the other types of ground tissue. So notice, whenever we see pith, our cortex, and it's also the cells that are found in the mesophyll of leaves. Whenever we see mesophyll, pith, cortex, we're mostly looking at parenchyma cells. So we want to keep that in mind. Now, another type of ground tissue, or it's sometimes called fundamental tissue or simple tissue, is collenchyma cells. And this is looking at probably corn again, it looks like. If we look, this is towards the edge of a stem. So this upper kind of curved area, this is the epidermis. If we look back in here, we see some parenchyma cells. Notice these little triangles and rectangles between the cells. Those are the spaces between the cells. The collenchyma cells are the cells in this region in here. Now keep in mind, they really are three-dimensional. Obviously, we can't see that too well here. So sometimes they're round, sometimes they're a little more oval, sometimes they're almost rectangular. But here's the key when we look at collenchyma cells. Again, keep in mind the dark purple is the cell wall. The cell walls in the nucleus tend to absorb stain. If we look at the cell wall, think about the width of a cell wall down towards the bottom in one of our parenchyma cells in here, and compare that to the thickness of cell walls up here. Well, there's two things that are pretty noticeable. The parenchyma cell walls are thinner. They're not as thick. But also the parenchyma cell walls are pretty much the same thickness all the way around. If we look at the collenchyma cell walls, some places they're really thick, like right in here. Some places right next to that, they're really skinny. So one of the things that we typically use to describe collenchyma cell walls is we say that they're thickened, but they're unevenly thickened. And collenchyma cells mostly provide support for plants. So wood can support a plant, but collenchyma is important in plants that don't make wood. It's especially important in leaves, even in woody plants. And it's important in something called the petioles. The petiole is that kind of stalk that attaches a leaf to the main part of the plant. And we want the petiole to have a little bit of rigidity to it so that it holds the leaf up into the sun. We don't want all the leaves to be drooping towards the bottom of the plant. So when we look at this, we see these unevenly thickened cell walls. This is a giveaway that we're looking at collenchyma. Now keep in mind, if we would look at a plant that has some collenchyma in it, there's much more parenchyma cells. There's a lot more of that type of tissue. So collenchyma is not as widespread. It's not as common as parenchyma tissue is. The third type of simple tissue, or it's also called ground tissue or fundamental tissue, is sclerenchyma cells. So if we look at this slide, what we see is up towards the top, those greenish thin lines, those are the cell walls of parenchyma cells. Notice that the cells themselves are relatively large. Again, we can see some little air spaces in between them, those little triangles, which is just a result of stacking kind of roundish oval cells together, like stacking a bunch of basketballs. You'd have gaps between the basketballs. If we look at the sclerenchyma cells, what we see is the cell walls are much thicker. So again, sclerenchyma cells have thicker cell walls than parenchyma do. But notice they're roughly the same thickness all the way around. They're not thicker in some locations and thinner in others, like the collenchyma are. Keep in mind that the cell wall is not just this kind of brighter orangish red line, but it's also that, that kind of deeper maroon red in the inside of each cell. So if you look at these cells, a lot of the cell is actually the cell wall. In fact, most of the volume of the cell is typically taken up just in the cell wall. So that's the main thing we can see. Now, when we look at parenchyma cells and collenchyma and sclerenchyma, one of the things we see is that some of these cells are associated with xylem or phloem. So when we talk about vascular tissue might have some parenchyma cells in it or some sclerenchyma. Usually doesn't have much collenchyma, but you know, if you're looking this up online to get more information, you might see that parenchyma called vascular parenchyma or vascular sclerenchyma, or it might just say parenchyma, because some people classify it as part of vascular tissue. Some people say, nope, it's just parenchyma cells in a different location. So we aren't going to worry about that. We'll let the botanists argue about that. So when we look at parenchyma cells, thick cell walls again. And whenever we see thick cell walls, keep in mind when we look at a microscope slide, those are usually going to be a dark color. So they're typically easy to identify from that way. And whenever we have thick cell walls, we always want to think of support, holding the plant up, whether it's collenchyma cells holding a leaf up so that the leaf doesn't droop, so that it's flat and collects more sunlight, or a stem, or even in some cases roots, because there are a few plants, even though we said roots are below ground, there are a few plants that have roots that extend above the ground. So these are the three simple tissues. So parenchyma is found associated with lots of other cells, but when we find parenchyma, we always find a bunch of parenchyma by itself. So that's why it's a simple tissue. When we find collenchyma, we always find a bunch of collenchyma by itself. So that's why it's a simple tissue. And similarly with sclerenchyma. Now, some tissues in the plant contain more than one type of cell. So we're going to look at one of these, which is called a dermal tissue. And dermal tissues are complex tissues. So in plants, if it's not a simple tissue, it's a complex tissue. And the difference is just simple tissues only have one type of cell. Complex tissues have more than one type of cell. So dermal tissues, dermal, again, think dermatologist means skin. Dermal tissues are on the outside of a plant. And the main thing dermal tissues do is they protect everything under them. If the dermal tissues get torn off, they can generally be replaced. Not necessarily true for the plants underneath it. If we look at most plants, and if we look at the root or the stem or the leaf, it doesn't matter where we look, the epidermis is the dermal tissue on the outside. In woody plants, depending on how long they live, the epidermis is sometimes replaced by periderm. Now, epidermis is generally one cell layer thick, so it's very, very thin. There are a few plants that have 2 or 3 layers, but they're in a real minority. And there are mostly 2 types of cells in the epidermis. Almost everything is in epidermal cells. And epidermal cells secrete a waxy cuticle onto the outer surface of the leaf. you could think of this cuticle as being like our sweat. And because it's waxy, this means it's a lipid-like material, which means it doesn't mix with water. So this is to waterproof the leaf or the other part of the plant, wherever it's secreting the cuticle. The other cells that we have commonly in the epidermis are guard cells. And guard cells surround holes in the leaf. These holes are called stomata. And guard cells let air in and out of the leaf so that the leaf can get carbon dioxide for photosynthesis. And also eliminate oxygen, because for plants, they use a little bit of oxygen for aerobic cell respiration. But since they make oxygen in photosynthesis, they make so much of it, they make more than they need. So what they can't use is released into the air. So water is also released through the stomata. So here's a section of a leaf where what we did was we peeled off the lower layer of cells, the lower epidermis. And what you can see is a bunch of cells. You can see the nucleus and the cell wall. Notice that these cells kind of all run in the same direction. The cells are all lined up nice and neat and parallel. Well, remember when we talked about monocots, we talked about the fact that they have those parallel veins, those ridges that you see on a blade of grass. So these are all epidermal cells. The guard cells are very thin. If you look very closely, the guard cells always occur in pairs, and they look kind of like lips. And if you want to think of guard cells as lips, the stomata is the opening, the mouth in between the lips. So we'll look later at the physiology of how guard cells can either open the stomata or close it. Now, why do we have to open and close it? Well, because we want to open it to let in carbon dioxide for photosynthesis. But when the stomata are open, the plant loses water. So it's kind of a balancing act. Do we have enough water that we can open the stomata, let some carbon dioxide in, and we can afford to lose that water? Or are we so short on water, we have to close the stomata so that we don't lose any more water, and we just won't be able to carry out photosynthesis for a while? And of course, in a plant, this isn't a thought process, this is a chemical process in terms of when the guard cells open and when they close. Now, one way that we can tell that this is a monocot, because if we look at all the pairs of guard cells and we look at the epidermal cells, they're basically parallel, like those veins that we would see in a leaf. Now, this is an example of a dicot epidermis. And remember that we said a dicot leaf would be a leaf like you would pull off a tree. So if you think about a leaf that you pull off a tree, there's that petiole, that main stalk that attaches the leaf to the tree. But once you get into the tree, then there are veins that branch out in different directions. So when we look at a dicot epidermis, we see that there are pairs of guard cells. Notice they're not all going the same direction. We only have 3 here, but if we had more, we would be able to see that the pairs of guard cells are aligned in all sorts of different directions. They're not all parallel to one another. And if we look at the epidermal cells, and notice this is an epidermal cell, this light green right around the pair of lips here. If we look at the epidermal cells, notice that the epidermal cells kind of look like, kind of some of those wild jigsaw puzzles where the pieces aren't nice and neat and even, and they all fit together. But notice, if you look here, you don't really see any 2 cells that are exactly the same shape. So if we're looking at a monocot epidermis versus a dicot epidermis, versus a monocot leaf versus a monocot stem versus a dicot leaf or stem or root, we can tell all these apart. So that turns out to be useful. Now, keep in mind, epidermal cells are a good example of how cells aren't necessarily the same everywhere in the plant's body. If we look at epidermal cells in the stem and in the leaves, or in other words, in the organ that we call the shoot, if we look at epidermal cells that are above ground, those epidermal cells tend to have a very thick cell wall. They don't have much cytoplasm. A lot of times they have a big central vacuole, and the central vacuole contains different toxins that poison, especially insects. That's the main thing that eats plants. They don't have any chloroplasts. So remember what we said before, the purpose of the epidermis is to protect the tissue underneath. So it's not really to carry out photosynthesis. But if you would look at epidermal cells in a root, they have thin cell walls. And the cell walls have hair-like processes. And these processes increase the amount of surface area that the plant has for absorption of water and minerals. So in the shoot, we're trying to avoid losing water. In the root, we don't want cells with thick cell walls. We don't want cells that have a thick waterproof coating. Why? Because we're trying to absorb water in those areas. Now, if we look at our guard cells and we see how they work, what we see is that if we look at a pair of guard cells with the stoma in between them, and the green represents the cell wall, what we see is that the cell wall in the middle, where the two guard cells meet, is thicker than the guard cell on the outside. Now, when the plant has enough water, so here's that central vacuole of the guard cell. When the plant has enough water, this cell absorbs water from the other parts of the plant. The water is put into the central vacuole. And when the water goes into the central vacuole, it causes the plant cell, the guard cell, to bulge. Now here's the thing, the cell wall on the inside is really thick. So it doesn't bulge as far as this thinner part of the cell wall towards the outside. So since the thinner cell wall bulges more, it kind of pulls this thick cell wall towards the middle. It kind of pulls that open, and that's how the stoma opens. Now, if the plant starts to run low on water, what happens? Well, when plants run low on water, usually the first part of the plant cell that loses water is the central vacuole. So the central vacuole loses water and shrinks. That water goes into the cytoplasm. That water can leave the guard cell, but even if it stays in, the cell is smaller now. That central vacuole, when it shrinks, it doesn't push these inner cell walls open. So they clump together and they close. So if the plant has access to a lot of water, the stoma will be open. If it doesn't have enough water, the stoma closes. But it's strictly a chemical, physical process. So we'll look at a little bit of the chemistry tomorrow, but we won't do that today. So let's look at the last type of dermal tissue, and then we're going to wait to focus on the other type of complex tissue, vascular tissue. So let's look at the periderm. But before we do that, we want to keep in mind that some leaves and stems do have another type of cell called a trichome that's found in the epidermis. And these trichomes make hairs and thorns and things like that that you've probably seen or felt on some plants. we don't typically find these structures underground, so it's usually on the part of the epidermis that's above ground. Now remember, the periderm is the bark outside the phloem, and it's the area that is going to replace the epidermis if we're talking about a woody plant. So we only find periderm in plants that have lateral meristems. And there are 3 types of tissues, or 3 types of cells, if you prefer, in the periderm. These 3 types of cells, this is why this is a complex tissue, why it's not a simple tissue. Because simple tissues, if we're looking at a clump of parenchyma tissue, it's all parenchyma. But here we've got 3 different types of cells. We've got cork cells, and cork cells are dead when they're mature. Now the cell wall has a lot of something called suberin in it. And suberin is hydrophobic. It's made up of long chain fatty acids. So remember fatty acids we saw in 105, they were some of the things that combined with glycerol to make a phospholipid or a triglyceride. And since they have a hydrophobic material, since they have a molecule that doesn't mix with water, what do cork cells do? They help prevent water loss. Remember, we've said on several occasions, plants lose 90% or more of the water that's absorbed by their roots. So this means that when we look at plants, conserving water is a big deal. So one way to conserve that is to have the outer part of your body covered with hydrophobic chemicals. Now the cork cambium is what divides by mitosis and makes cork cells. Sometimes it makes another type of cell called phelloderm cells to the inside. Phelloderm cells are thin layered parenchyma cells. And phelloderm cells are alive. Parenchyma cells are always alive. So here's what we could see if we looked at a typical plant. So notice the outside of the plant is to the top. So early in the plant's life, if we look at the periderm is pretty thin. We have this dark brown layer, which represents the cork cambium. We have a layer of cells called the phelloderm to the inside. And we have a layer of cells called cork cells to the outside. And then we have the epidermis above that, the dark green. And we have the kind of olive green, the cuticle. And remember what we said a few days ago, the cuticle is not cells, it's waxy chemicals. So waxy chemicals, again, lipids, hydrophobic, waterproofing. Down below, we see what would happen if this periderm grows for a period of time. So notice the phelloderm is generally only 1 cell layer thick. There are a few weird plants where it's 2 or 3, but they're pretty weird. The cork cambium is still only 1 cell layer thick. The cork, notice there's now 3 layers of cells. And at this point in the plant's life cycle, we still have the epidermis and we still have the cuticle. But if we follow this plant as it grows further, as we get 4 or 5, maybe 10 layers of cork cells, this cork eventually, notice it's pushing the epidermis further and further out from the plant. And eventually these epidermis cells just can't stretch anymore. And so the epidermis tears. And eventually, if we kept following this, we could find kind of flaps of epidermis hanging off the outer edge of the plant. But most of the bark that we would be looking at would be this cork. So the other type of complex tissue is vascular tissues. and we will look at them tomorrow. Are there any questions before we leave today about the tissues we've talked about or anything else that comes to mind? Okay, if not, then I'm going to stop screen sharing. And if you have any questions, feel free to ask them before we go, but otherwise, I will see you tomorrow. Don't forget the animal questions are finally due today. I got the right day, thanks to Ian.
Have a good day.
You too, Jade.
Have a good day.
You too. Enjoy our lovely sunny afternoon. Hey, we had a few good days. We can't complain. It is Chicago.
Mm-hmm.
Yeah.
What did we expect?
Have a good day. See you Friday.
You too. Bye-bye.
Bye.
Bye, Mr. Griffin.
Bye. See you tomorrow.
Mr. Griffin,
Hmm.
will you be available today?
I might be, but probably not till between about 3 and 5 because I have meetings until then, and then I have a meeting in the evening.
Yeah, what about 5 o'clock?
Well, we probably have to finish by about 5.
Okay. Oh, we have to finish by 5?
Yeah.
Mm,
Yeah. Send me an email and let me know if you can meet during that time frame later today.
wait, you'll be available by 5 o'clock upwards? Or,
Yeah, I could maybe stay till about 5:30, but probably not much later than that.
okay, thank you.
You're welcome. And keep in mind, Ian, I wasn't being sarcastic when I said I finally got the date right because of you. That's the truth.